In this next lecture, we will start to transition away from the diptera. Although flies are very important in forensic entomology, other insect groups, such as the beetles, also play a role. This lecture is going to consider the topic of forensic entomology and will contain some pretty graphic images. You have been warned. Forensic entomology is the use of insects that inhabit decomposing remains to aid legal investigations. Urban forensic entomology typically concerns pest infestations in buildings or gardens that are the basis of litigation between private parties and service providers such as landlords or exterminators. Stored product forensic entomology is often used in litigation over infestation or contamination of commercially distributed foods by insects. The forensic entomologist may serve as an expert witness during both criminal and civil proceedings involving food contamination. But typically, when we think of forensic entomology, we think of the medical legal, medical criminal aspects, and that's what we're going to focus on. Medical legal forensic entomology includes arthropod involvement in events such as murder, suicide, rape, physical abuse, and contraband trafficking. Insects can be used to determine the post-mortem interval, or PMI, and the location of the death in question. Since many insects exhibit a degree of endemism, which means they occur only in certain places, or have a well-defined phenology, meaning that they're active only at a certain season or time of day, their presence in association with other evidence can demonstrate potential links to times and locations where other events may have occurred. For example, an Ohio man who claimed to have been in Ohio on the date his wife and children were murdered in California was found to have grasshoppers and other nocturnal insects from the west on his car grill, indicating that the car had been driven at night to the western U.S., and he was subsequently convicted of these murders. Another area of medical legal forensic entomology is the relatively new study of entomotoxicology. This particular branch involves the utilization of entomological specimens found at a scene in order to test for different drugs that may have possibly played a role in the death of the victim. Insects are valuable as forensic indicators in cases of abuse and neglect. Some insects, such as the green bottlefly, are drawn to odors such as ammonia, which can result from urine or fecal contamination. Adult green bottleflies tend to be attracted to an incontinent individual who lacks a voluntary control of excretory functions. Such examples include a baby who has not had its diapers changed often, or an incontinent elderly person who has not been helped in maintaining routine bodily hygiene. Flies lay their eggs in and around clothing and skin, which, left undiscovered, will hatch into maggots, which begin feeding upon flesh, open wounds, ulcers, and any natural bodily entry point. Over time, the flesh will be eaten away, and the region may be further infected by bacteria or invaded by other kinds of insects. When a body is found, it's important as part of the investigation to determine the time of death. And there are a number of ways that this can be done. We can look at livor mortis, or the death color, the death chill, death stiffness, stomach and intestinal contents, decomposition, and then, of course, insects. So let's go through these. Death color, or livor mortis. What happens is that as the body begins to decompose, the blood is going to seep down through the tissues and settle in the lower parts of the body. In other words, just a response to gravity. And they refer to this pooling of blood as lividity. The RBCs turn a purplish blue. You can see that in the picture. This will begin about 2 hours after death. Between about 2 to 8 hours, the lividity will be present. But if the skin is pressed, the color will disappear. After 8 hours, the lividity will remain even if the skin is pressed. Higher temperatures increase the rate of lividity development. Lower temperatures decrease it. Other things can be determined from the death color or lividity as well. For instance, the position of the body following death, and whether or not a body might have been moved post-mortem. The temperature of a body can be used to estimate time of death during the first 24 hours. The core temperature falls gradually with time since death and depends on factors such as body mass, fat distribution, and ambient temperature. If the body is discovered before body temperature has come into equilibrium with the ambient temperature, then a forensic scientist can estimate the time of death by measuring the core temperature of the body. Now keep in mind that we're talking about the core. They have special thermometers that are long and very sharp, and usually they'll insert it into the liver, into the middle part of the person's body. And other kinds of factors can impact algor mortis as well. So the temperatures, the external ambient temperatures will play a role, but also things like whether the individual's clothed or not, or wrapped in a blanket, or factors such as that. But for the first 24 hours, it can provide a pretty good guess. Within about 2 hours after death, muscles start to stiffen. We call this rigor mortis. It begins with the small muscles and reaches its peak at about 12 hours after death. Within about 15 hours post-mortality, the muscle fibers start to break down and soften, and the stiffness will gradually disappear after about 36 hours. The reason for this stiffness is the physiology of the muscles themselves. Muscles are made up of two kinds of fibers, actin and myosin, that lock and unlock during muscle contraction and relaxation. These are the thin and thick filaments. These connections are controlled by a biochemical pathway within the cell, which is partially driven by the presence of calcium ions. When a muscle contracts, calcium ions are released from storage in the sarcoplasmic reticulum into the sarcoplasm of the muscle cell. ATP is also required. To relax, the calcium ions must be actively transported back into the sarcoplasmic reticulum, and ADP is resynthesized into ATP. After death, of course, aerobic respiration ceases, but anaerobic processes can continue. Eventually, ATP runs out, and the calcium ion gradient cannot be maintained. The muscles remain contracted at that point. The joints will remain stiff for 1 to 3 days, but after this time, general tissue decay and leaking from the lysosomes of their digestive enzymes will cause the muscles to relax. Rigor tends to start in the small muscles first, especially those in the face. That's why it's been ritually important to close the eyes quickly on someone who's died, because they're the first to become rigid in rigor mortis, and it was thought that a corpse with open eyes posed a threat to its kin. As has long been the case in many cultures, they used coins to keep the corpse's eyes closed. The practice of using coins endures, representing a feeling that money, so important in life, may also be important in death. A number of factors affect the rate of rigor mortis. Ambient temperature is one. Cold, of course, is going to slow the process down, and it'll speed up when the temperatures are warmer. Clothing also has an effect. If there's no clothes, that will slow things up. Clothing, because it retains the heat, accelerates rigor. Sun exposure, activity or exercise also plays a role. If somebody was exercising or struggling at the time of their death, that will accelerate rigor. If they die during their sleep, the development of rigor tends to be slow. And body weight also plays a role. When someone is overweight, that slows rigor. Someone who's very thin will see the rate of rigor much faster. Another category used to estimate the time of death is the stomach and intestinal contents. If the stomach contains undigested food, then death occurred within 2 hours after the last meal. If the stomach is empty but food is found in the small intestine, then death occurred probably 4 to 6 hours after the last meal. And if the small intestines are empty and wastes are found in the large intestine, then death probably occurred 12 or more hours after the last meal. And then, of course, there's decomposition. With decomposition, we're talking about the rotting or breakdown of tissues and organs. Bacteria and other microorganisms aid this process, just as they do with the decomposition of plants and animals in the environment. And decomposition occurs in a predictable pattern, and this can be used to determine the time of death. We'll look at the different stages of decomposition later. But this brings us to the question, why do we use insects to estimate the time of death? Why are they used forensically? Well, that's because insects are usually the first to discover a corpse. If a death occurs outdoors, blowflies may arrive and start laying eggs on the corpse within a mere minutes after the person has died. And another aspect to the use of insects is that insects arrive at a decomposing body. So first, which insects are important? Well, flies are probably the most important. Calliphorids and so on, the blowflies, the flesh flies, house flies, all play a role. Soldier flies, coffin flies are important. But beetles, such as carrion beetles and rove beetles, dermestids, are also very useful. And then other insects like ants and earwigs and cockroaches can all play a role. But first, let's look at flies. Okay, so with the flies of forensic importance, the caliphorids and the sarcophagids are the two families that are prominent. Now, maggots, the larvae of flies, are the most obvious and abundant fauna present on corpses in the early stages of decomposition. Houseflies and blowflies are the first to arrive. They're what we call pioneer flies. Flies in both of these families lay eggs on the corpse. The recently hatched larvae, as well as their parents, initially feed on the fluids that exude from the body. Later, they enter the body through natural openings or wounds and eventually feed over the whole body as tissues decay. Different species of flies arrive at different times after death. So as we continue, the juicy maggots that are there from the blowflies and the sarcophagids provide an abundant food source for other types of animals and other types of insects, including other species of flies. There's a species of blowfly that feeds on the maggots of other flies, as well as consuming dead flesh. These are the chrysomya. The chrysomya larvae are covered with protrusions called papillae, which serve as protection against the predatory attacks of other maggots. Now, when the corpse is dried out, two other groups of flies, the cheese flies and the coffin flies, join the beetles and mites in cleaning up the skeleton. That brings us to the beetles. The first beetles arrive at a corpse soon after the body begins to putrefy. In contrast to the flies, beetles have chewing mouthparts and can manage tougher foods than the semi-liquid material that fly larvae are so efficient at exploiting. Three types of beetles make their living out of corpses. The early arrivals tend to be predatory adults that feed on fly larvae. Some of these species lay their eggs in the corpse, and the emerging larvae, which share their parents' powerful jaws, also feed on fly larvae. These species include the rove beetles and the hister beetles. Late arriving species tend to be specialist scavengers, which feed on tougher parts like skin and tendons as the body dries out. The dominant late-stage scavengers include the larvae of hide beetles, or dermestids, and ham beetles, the clarids. Species such as the carrion beetles, the sylphids, are more variable in their diets. The adults are tend to be predatory, although they will eat some carrion, but their larvae are restricted to carrion and moist corpses. And these next few slides, we're going to be looking at how do they do an investigation when a dead body is found. And of course, we're going to be primarily concerned with how we use insects and determining the postmortem interval. Okay, so first, collecting the insects. What do we need to collect? Well, basically all of the insects, the adults and the immatures, also insect parts, because you may find the skin of puparia if you've come upon the body late enough that some of the flies have completed their development. Specimens should be collected from the body, also under the body, and from the surrounding area. Keep in mind that the higher fly larvae, when they reach the prepupal stage, the third instar larva stops feeding and it tends to migrate from the breeding site to a drier place where it's going to pupate. So you need to look at insects and collect from around the surrounding areas as well. The proper collection of insects may require the use of aerial nets and forceps, and you want to take both live and preserved specimens. So once you've brought maggots in, oftentimes some of the samples are going to be reared to the adult stage. This is important because it's much easier to make a species identification from the adult fly than it is from a maggot. So typically we'll collect 20 to 40 large larvae, place them in several rearing cups. We'll repeat the procedure with smaller larvae and eggs. And then they'll put them into a cup. You can see there's some meat in a foil container there. That's minced up pork loin. That's generally what is used for feeding the larvae. It's a good stand-in for. When you're collecting the flies, you notice various kinds of patterns. Large numbers of eggs are usually placed around the eyes, the nose, and the mouth. Blowflies are usually the first to come in, and open wounds or trauma sites are particularly attractive as oviposition sites. So this can give some insight to an investigator as to where the corpse may have been injured. Sometimes later in decomposition, it isn't always real obvious where various. So additional data, of course, you want to get visual observations. You want to write down everything about the site, what the habitat was like, the location and orientation of the body. Was it exposed, covered, clothed, and so on. You want to make note of any obvious trauma that you see, record any damage to the body, and you also want to make a record of any evidence of scavengers having fed on the corpse. You want to note the locations of major insect infestations, you want to get a GPS reading, and you want to get weather data. Additional data collection would include temperature. And with temperature, you of course want the ambient air temperature, which we take 4 feet from the top of the soil in the shade. But you also want body surface temperature, ground surface temperature, soil temperature, and the maggot mass temperature. You see the arrow pointing to the maggot mass. As the number of maggots gets really, really high and very dense, their activities generates a certain amount of heat too, and you want to get a temperature reading from that. And this picture just shows you all the different kinds of things that you're going to want to collect if you're doing an investigation. And notice one thing that they keep indicating on this slide: labels. Very careful data has to be taken because remember, these are going to be perhaps part of a court investigation, and the forensic entomologist will be asked to participate as an expert witness. And you have to have all your data in a organized in a book. All of your samples have to have been well labeled and so on, because any mistake that you make, any sloppiness that you show in your methodology will be used by the defense attorney to to question your competence. Okay, so how do we estimate the postmortem interval using insects? Well, there's 2 main ways that we can do it. One is based on insect development. Of course, this is based on the fact that insects tend to have a pretty constant rate of development at a particular temperature. So we can use this knowledge to determine the postmortem interval. But we can also use insect succession. An examination of the insect species present or absent makes a difference. Remember, I said that the body is going to change over time, not only because of decomposition, but because of the feeding activities of the insects that are there. So the early flies that are coming in, the pioneer flies, make changes from their feeding to the nature of the corpse that makes it now suitable for other kinds of insects. And so this leads to a succession, and this succession is pretty predictable. So this can also be used. But let's look at these in more detail. So first, insect development. This method applies the larval development times to investigations. During early decomposition, entomological estimates of postmortem interval depends on the time period required for insect species represented to develop to the growth stage that was collected at the death scene. Most often, the larvae of the flies belong to the caliphorids and the sarcophagids. These flies usually provide the most accurate estimate of time of death. The age of specimens collected from the victim may be estimated to provide a minimum time period since death. This approach requires the detailed knowledge of the fly species used and the conditions at the crime scene, but it is relatively conservative. It is mentioned that because of their relatively short. So if we're going to work up a case, remember, you have to collect specimens, obtain temperature data, and now you want to collect, or calculate, I should say, the ADH for time period. So another method of calculating postmortem intervals involves estimation of the accumulated degree days or hours, or ADD or ADH. ADH values represent a certain number of energy hours that are necessary for the development of insect larvae. The degree day or hour concept assumes that the developmental rate is proportional to the temperature within a certain species-specific temperature range. However, the relationship between temperature and development rate is typically curvilinear at high and low temperature. So, uh, again, to use insect development, you have to know very precisely the development times of the insects that you're using. So, this is the kind of data that could be collected for the bottlefly, Phoenicia sericata. So there's a minimum development temperature, in the case of this species, 10 degrees centigrade. If it's cooler than that, then they stop developing. Then you have an environmental temperature, you measure what that is, and then you can do calculations of total development time and calculate your degree hours. So we would do studies on these flies. So here you can see that at 70 degrees Fahrenheit, this is the length of time it takes for this species to go to each of these different developmental stages. Now, this kind of calculation is only going to be accurate, though, at 70 degrees. If it's 75 degrees, then you're probably not going to be accurate, or if it's cooler than that, that affects your model. So typically, laboratories that are doing these kinds of studies are going to get the data rearing their flies at a number of different temperatures. Okay, so to finish up on using insect development as a method for estimating time of death, just remember there's some basic considerations. First, the use of ADH and ADD is based on the assumption of a linear relationship between temperature and development. And this assumption is only going to be valid when the data used to generate the degree days are close to the temperature at which the degree days are applied. When you get to extremes, linearity is lost, meaning that at very high temperatures and very low temperatures, this relationship is no longer valid. And it's best to use developmental data that was generated within plus or minus 5 degrees centigrade. So as I said before, we typically will see laboratories that are routinely doing forensic analyses raise flies at a variety of temperatures so that they have very extensive data on development time and can pick the right temperature to do their calculations. The principle of insect succession can be used to estimate the PMI. Different species of insects prefer different stages of decomposition, As the body decays further, it becomes less favorable to early colonizers and more desirable to other species. Studies have been conducted using animal carcasses and human cadavers to develop models for succession in a variety of climates and regions. Out in Tennessee, I believe they have a facility called the Body Farm, where they put out bodies that have been donated to science and develop models for estimating these kinds of succession rates. This allows researchers and forensic investigators to make reliable PMI estimates even for severely decomposed bodies. Many factors can influence the rate of succession and decomposition. The spread of chemical breakdown in the body and the rate of growth for young insects are highly dependent on ambient temperature. Decomposition and insect growth will be faster in warmer climates, while cooler temperatures may retard or even prevent insect development. The time of day can affect how quickly the body is initially colonized, and the season can affect which types of insects will be present throughout succession. Whether the body is covered, wrapped, buried, submerged, or enclosed in a small space can all alter the insect community. It is also important to consider the body's surroundings. If there were other sources of carrion feeding insects nearby, such as a garbage dump or even an animal carcass, insects may migrate to the new body earlier than if the body was the original rearing site. This possibility has been used in court cases to contest the validity of a forensic entomologist's report. Both in the lab and in the field, forensic entomologists must take great care to note all possible evidence which may have an impact on the investigation. This slide shows you the basic succession of insects. So on the left side, we have the condition of the body as it moves through different stages of decomposition, and on the right, we have the types of insects that you're going to find on the body at that time. So when the body is fresh, the pioneer flies are going to be there, the blowflies, the flesh flies, and so on. Then when the body moves into the bloated stage, the flies are going to give way to some of the beetles, such as the rove beetles, scuttleflies, coffin flies. Then, when it reaches the stage of active decay, we still have flies, but cheese skippers, rove beetles, and carrion beetles are going to be much more prevalent. Then, in advanced decay, it's rove beetles. So here, when you're going to use succession, you need to get baseline data. You need to get baseline fauna that's obtained by doing controlled experiments. When a body is found in the same or similar area, then the corpse can be compared to it. And as I mentioned, there are places around the country where bodies have been donated to science to do these kinds of studies. But in many other areas, they use a pig carcass as a stand-in for humans because So we had a graduate student who did a study on forensic entomology. And this is an example of the kind of data that she got. So you can see all the different sorts of taxa that she collected. And the sampling interval across the top, those are just days. So she had a pig carcass put out, and over a series of days, she went out and collected the insects from that. And you can see on the left-hand side, you've got the early pioneer species that then drop off after you get into the later stages of decomposition. Okay, so now we're going to look at these different stages of decomposition and examine the different species of insects that come in. And as we do this, you're going to see that in addition to the physical changes of the body, there's also differences in the volatile compounds that are given off by the body. Two of the most notable would be cadaverine and putrescine. People who work in law enforcement will often mention how you walk into a room and you can know before you even see the body that there's a dead body there. And that's because of these types of volatile compounds that are given off. So if we look at the stages of decomposition, the first stage, of course, is going to be initial decay. So although the body shortly after death appears fresh from the outside, the bacteria that before death were feeding on the contents of the intestine begin to digest the intestine itself. They eventually break out of the intestine and start digesting the surrounding internal organs. The body's own digestive enzymes, which are normally in the intestine, also spread through the body, contributing to its decomposition. On an even smaller scale, enzymes inside individual cells are released when the cells die. These enzymes break down the cell and its connections with other cells. Then we have insect activity. From the moment of death, flies are attracted to bodies. Without the normal defenses of a living animal, blowflies and houseflies are able to lay eggs around wounds and natural body openings, such as the mouth, the nose, and the eyes. These eggs hatch and move into the body, often within 24 hours. The life cycle of a fly from egg to maggot to fly takes from 2 to 3 weeks. It can take considerably longer at cold temperatures. Then we move into putrefaction. So the bacteria continue their work. They're breaking down the tissues and cells and releasing fluids into the body cavities. They often respire in the absence of oxygen, that is to say, anaerobically, and produce various gases, including hydrogen sulfide, methane, cadaverine, and putrescine as byproducts. People might find these gases foul-smelling, but they're very attractive to a variety of insects. The buildup of gas resulting from the intense activity of the multiplying bacteria creates pressure within the body. The body inflates or bloats and forces fluids out of cells and blood vessels and into the body cavity. In terms of insect activity, the young maggots move throughout the body, spreading bacteria, secreting their own digestive enzymes, and tearing tissues with their mouth hooks. They move as a maggot mass, benefiting from communal heat and shared digestive secretions. Remember that picture that we looked at earlier of a maggot mass. The rate of decay increases, and the smell and body fluids that begin to emanate from the body attract more blowflies, flesh flies, beetles, and mites. The later arriving flies and beetles are predators, feeding on maggots as well as the decaying flesh. They are joined by parasitoid wasps that lay their eggs inside the maggots and later inside pupae. So now we move into black putrefaction. The bloated body eventually collapses, leaving a flattened body whose flesh has a creamy consistency. The exposed parts of the body are black in color, and there is a very strong smell of decay. A large volume of body fluids drain from the body at this stage and seep into the surrounding soil. The other insects and mites feed on this material. The insects consume the bulk of the flesh, and the body temperature increases with their activity. Bacterial decay is still very important, and bacteria will eventually consume the body if insects are excluded. By this stage, several generations of maggots are present on the body, and some have become fully grown. They migrate from the body and bury themselves in the soil where they become pupae. Predatory maggots are much more abundant at this stage, and the pioneer flies cease to be attracted to the corpse. Predatory beetles lay their eggs in the corpse, and their larvae then hatch out and feed on the decaying flesh. Parasitoid wasps are much more common, laying their eggs inside maggots and pupae. At this stage of decomposition, all the remaining flesh is removed and the body dries out. It has a cheesy smell caused by butyric acid, and this smell attracts a whole new suite of corpse organisms. The surface of the body that is in contact with the ground becomes covered with mold as the body ferments. The reduction in soft food makes the body less palatable to the mouth hooks of maggots and more suitable for the chewing mouthparts of beetles. Beetles feed on the skin and ligaments. Many of these beetles are larvae. They hatch from eggs that were laid by adults, which fed on the body in earlier stages of decay. The cheese fly consumes any remaining moist flesh at this stage, even though it is uncommon earlier in decay. Predators and parasitoids are still present at this stage, including numerous wasps and beetle larvae. The body is now dry and decays very slowly. Eventually, all the hair disappears, leaving bones only. Animals that feed on hair include various kinds of moths, microorganisms like bacteria. Mites, in turn, will feed on these microorganisms. They remain on the body as long as traces of hair remain, which depends on the amount of hair that covers a particular species. Humans and pigs have relatively little hair, and this stage is short for these species. Okay, so in summary, forensic entomology is based on insect attraction to and interaction with human remains. It's a very useful tool that's been known and used for centuries in medical criminal investigations, and it provides an alternate approach to estimating postmortem intervals. It's an expanding field, but we have to recognize that it has a number of constraints and may provide inaccurate information unless great care is taken.